And I would just look here, use the pointer here.
Use the pointer.
Yes, just right here. And use this.
Oh, I need to keep it. Yeah, I think it is better if you use this one. So that way. Okay.
No, don't use that.
Oh, from the previous talk, we see that e-cigarette is more and more popular, but we don't know. We don't have enough information for it's good for our health or bad. We need to do more research. Yeah, e-cigarette has a variety of different products. They have different shape, different ingredient in the e-liquid, and even different mechanism to activate. To study the health effect, we need to have animal models, but rats or mice, they don't smoke, so that's the necessity. We need to have animal model, and we need to have equipment to study e-cigarette effects. Recently, NIDA designed a standardized research e-cigarette for the purpose that our research can be more consistent. We can compare different results from different labs. So NIDA designed the dimension of e-cigarette, designed the ingredient in the e-liquid, and even have specification of the discharge battery and how big is the volume of the e-liquid in the tank. So for nicotine delivered to animals, for drug abuse study with animal models to be relevant to human, we need to have the route of drug administration similar to human drug users, and the blood pharmacokinetic or tissue concentration similar to human, equivalent to drug users. So, for e-cigarette, we need to understand two concepts: vapor and aerosol. The e-cigarette industry wrongly claim that e-cigarette is nicotine vapor. Actually, it's not. It's aerosol. A vapor is a gaseous state of substance. Aerosol is suspension of small particles in air, like tobacco cigarette or e-cigarette. Many studies have shown that there are particles in the e-cigarette smoke. The size of the particle are similar to conventional cigarettes. So the mechanisms are different. Conventional cigarette burning tobaccos, e-cigarette heating the e-liquid that contain propylene glycol or glycerol. So aerosol get to the lung, the deep lung, but the vapor deposited in the in the mouth, in the nose, and upper airway do not get to the lung. So they're different in the concentration and different area of absorption in our body. Nicotine aerosol, like e-cigarette, can reach high nicotine concentration in the air compared to vapor. Aerosol particles carry the nicotine into a villi region of the lungs. So nicotine in pulmonary membrane get into the circulation quickly into induce cardiovascular effect within 3 to 4 seconds. So there are studies showing pharmacokinetic after smoking e-cigarette. You see in different dose of e-cigarette, there are different mg of concentration in the e-liquid. So the higher nicotine, you expect a higher concentration in the blood. So the nicotine administration in vivo animal model, the rapidity of absorption, quick rise in the peak blood nicotine concentration play a role in determining the magnitude of this effect, especially cardiovascular and pulmonary effects. Slowly rising nicotine concentration result in desensitization without activation, and some nicotine receptor characteristics. So we start develop a non-invasive method deliver e-cigarette also to rodent models through inhalation. In the briefing zone of this system, the aerosol droplet size distribution are close to what e-cigarette smoker inhale into their respiratory system. Here you see this picture before. Here is the chamber the mice put in here, and here is the holder of e-cigarette. E-cigarette actually open to the chamber and to this animal. So there is the activation valve. When the valve open, pressurized air get into the e-cigarette and then generate e-cigarette smoke in the animal chamber. This is a control unit. We can control the timing and duration of each e-cigarette path. We can define or program this equipment to activate e-cigarette, the timing of different paths and different waiting cycle and the time during the day. So with some preliminary data, we test the blood concentration of nicotine in mice, and we get concentration in the blood, nicotine concentration in the blood close to what we see in human vapors. So mouse e-cigarette chamber generate nicotine and cotinine levels in the range of human smokers and e-cigarette smokers. The method and device tools for study e-cigarette smoking on cardiovascular system, respiratory system, nervous system, addiction, metabolism, development, and so on.
Thank you, Max.
Thank you, Dr. Shao, for developing the chamber. Okay, with this. Yeah, okay. Yeah. For developing the chamber. Now, what I'm going to do, I'm going to show you some of the adverse effects of e-cigarettes on liver and heart. Now, in order to do so, we used FOE. We used FOE mice. As these mice, even when fed a Western diet consisting of 20% protein, 50% carbohydrate, and 21% fat, and 0.21% of cholesterol, as opposed to very high fat diet, are prone to develop hepatic steatosis. Furthermore, because non-alcoholic fatty liver disease has been identified as an independent risk factor for atherosclerosis and cardiovascular disease, an additional advantage of this model is that it would be an ideal model for linking obesity, atherosclerosis, and hepatic steatosis. Accordingly, FOE null mice were exposed to ENS or saline aerosol for 12 weeks. At this point, we purposely used shorter duration to examine the detrimental effects of ENS on the initiation of NFL, because a longer exposure to Western diet alone results in hepatic steatosis. Okay, here you see as expected from expected from IP injections of nicotine, we also see ENS shows ENS also produce a modest reduction of body weight and cumulative food intake, but it increases locomotor activity. Here you can see ENS, when combined with the Western diet, causes hepatic steatosis. Now, we use high resolution light microscopy involving glutaraldehyde fixed, osmium tetroxide, post-fixed, epoxy embedded, and polyethylene blue stain sections to detect lipid. Now, glutaraldehyde fixation, osmium tetroxide, post-fixation allows retention of the lipid that would have normally been washed out during the process, clearly as opposed to controls. ENS exposed FOE null mice showing higher accumulation of lipid or hepatic steatosis. We further performed electron microscopy to show in greater details of lipid accumulation compared to control, where you can see normal architecture. You can see lots of lipid accumulation inside hepatocytes. Also, in this slide, there is an apoptotic hepatocyte nucleus. We next put our attention to effect of ENS on heart. And here, cardiomyocytes form a normal, from saline exposed FOE null mice showing normal nucleus, as well as sarcomeric and myofibrillary architecture, as opposed to control mice. Cardiomyocytes from ENS exposed mice show varying degrees of nuclear as well as cytoplasmic abnormalities indicative of cardiomyopathy and contractile dysfunction. So clearly, the prominent nuclear abnormalities included condensation and fragmentation of chromatin, and also you can see the abnormal pattern, very convoluted nuclear membrane, and the prominent cytoplasmic abnormalities also included. This is very important. Also included thinning of myofibrils, destruction, and as well as complete dissolution. So you can see how compared to this, how this myofibrillar thinning. The other cytoplasmic abnormalities included lipid. Lipid accommodation juxtaposed to intramyofibrillar mitochondria as well as mitophagy. So in conclusion, AINS, when combined with a Western diet, leads to hepatic steatosis in APOE null mice. AINS in combination with a Western diet can also cause ultrastructural changes in the heart ventricles, indicative of cardiomyopathy and contractile dysfunction. We surmise that AINS plus Western diet is likely to be a very toxic combination in patients with detrimental effects in both liver and heart. Thank you.
One per person.
Okay.